Hello, greetings from Qatar. It's an honor to be sharing some of my experiences and research work in the laboratory of Professor Namjoon Cho in this forum today. First of all, I would like to thank Professor Cho for inviting me for this online presentation. Thank you, Professor Cho. Let me begin by saying that I moved to Singapore in 2011 and joined the Mechanobiology Institute as a research fellow and worked there with Professor Jay Groves for a while before moving to the Nanyang Technological University in the laboratory of Professor Namjoon Cho. Uh, I had a extremely fulfilling experience in the laboratory of Professor Cho, working on some of the aspects of lipid bilayers and their assembly, and using those lipid bilayers for biological studies. And this is what I'm going to share with you today in a little bit more detail. On the right side, you can see a picture of a part of the laboratory, which was we were sitting at the uh, fifth floor of the Research Technoplaza building, and we're having fun. So let me give you a brief idea about the cell membrane, which is what is the focus of our today's presentation. Cell membrane is made up of a lipid bilayer, and the key issue is that it's a very physically complex environment. And as you can see, there are different kinds of proteins, other biomolecules, and the cytoskeleton inside. And this gives rise to many interesting phenomena that we would like to explore. So for this, we use supported lipid bilayers. So these are synthetic bilayers, which allow assembly of uh, uh, assembly of molecular clusters that resemble those that form at the real cellular system. Shown here is a schematic of how supported lipid bilayers are formed. We see these vesicles on a hydrophilic glass surface, and based on the physicochemical properties of the surfaces, they either form these planar lipid bilayers, or they remain as vesicles in other conditions. This is some of the work that I did at the laboratory of Professor Cho. We can see that with increasing pH, we lose the ability of these various combination of lipid vesicles in their ability to form supported lipid bilayers. Here is another piece of work that I did. I was fortunate enough to be a part in the laboratory of Professor Joe, where we look at the binding of this protein called monoamine oxidase. It's an important enzyme involved in the neurotransmitter metabolism. You can see that these two proteins, which one is a monomer, the other one is a dimer, can interact with lipid bilayers in a charge-dependent manner. More on how we use these supported lipid bilayers. So we specifically use these supported lipid bilayers to study cellular signaling events. These lipid bilayers mimic cell membrane and allow some assembly of receptor ligand clusters that can induce signaling inside the cell. The reason we use supported lipid bilayers is that we can control the physical as well as chemical properties of the membrane, as well as the biomolecules that we display on the membranes. Here is some example of schematic of a supported lipid bilayer. We can put fluorescent molecules on the lipid bilayer. We can attach fluorescent proteins on the bilayer to monitor the assembly of the bilayer. And of course, we can create these kind of different patterns. So this is a planar bilayer. This is with a grid, metal grid pattern. These are nano pattern substrates. We can create membrane microarrays, micro pattern substrates, where we have small islands of lipid membrane. And then we can also make lipid membranes displaying different proteins in a spatially segregated manner. This is an example of an image showing a lipid bilayer which is fluid and it covers fluorescence within a matter of 2 minutes when fluorescence recovery after photobleaching experiments are done. On the other hand, this is a viscous bilayer which do not recover its fluorescence intensity. So in a way allow us to control the physical properties of the bilayer. Some of the key discoveries we have made is that the, there's a cell adhesion protein called E-cadherin that forms adhesion in the epithelial tissue. We found that this protein, when it is highly diffusive, largely fails to form adhesion like the one shown here, as is less than 1% of the cells can form adhesion. Whereas if we reduce the mobility of the membrane, the this protein becomes competent to form adhesion very frequently. Now this is something that we found to be unique for this receptor and appears to defy all laws of chemical reaction rates. But this is what we discovered is that this process of cell adhesion assembly on supported lipid bilayers goes through a process of kinetic nucleation. Additionally, we found that the clustering of the protein on the cell membrane is a key regulator of mechanical signaling from the receptor. Shown here is a grid pattern substrate onto which a cell is forming adhesion. Creating these nano pattern grids, we can control the density and the assembly of clusters. As you can see outside the pattern, there's a lot of these proteins that can assemble, whereas inside there is limited number of molecules that can assemble. Here we are looking at the activation of a mechanosensitive protein called alpha-catenin. Application of force results in its opening, a conformational change that allows it to now bind an antibody called alpha-18. So what we have done here is that we change the pitch of the grids on the surface. So the 2-micron grids are large enough to allow assembly of large clusters. But as we go down to 500 nanometers, the size of the clusters reduces. And what this results in is the relatively less activation of alpha-catenin as shown from alpha-18 antibody staining. So we see that there is a correlation between the size of the E-cadherin clusters and the relative levels of activation of alpha-catenin. With this, I would like to conclude that lipid bilayers are an excellent material platform that enable a variety of cellular functions that we often are interested in. Supported lipid bilayers can capture many of the features of the cell membranes that are seen in living organisms, and we have successfully utilized these for understanding several biological systems, including the cell adhesion receptor that I just shared. And we continue to use these supported lipid bilayers for understanding and continuing our studies related to cell receptors in the future. Thank you.